Hello, everyone, and welcome to the third talk in the talk series hosted by the Association of Indian Primatologists. So, today's talk is by Dr. Krishna Balasubramaniam on social networks. But before the talk, let me just take a minute to talk about the association. The Association of Indian Primatologists was formed by students of primatology from India. So the main idea behind forming AIP was that to increase interaction among the Indian primatologists and to increase within country collaborations. So we have given the website link at the bottom of this YouTube video. So please visit the website and please like this video, subscribe to the channel and spread the word. And please click on the bell icon for notification. Always wanted to say that. So today's speaker is Dr. Krishna Balasubramaniam. He is at University of California, Davis, and uh, he's going to talk about social network analysis and its utilization in the field of primatology. So over to Krishna now.
Hello, can everybody hear me?
Yeah.
Okay, great. Thank you so much for hosting this AIP. And it's a pleasure to be here to talk to you guys today about a topic that's broadly of interest to a number of students as well as researchers around the globe. So I'm just going to be sharing my screen for a second and then we'll get going. Okay, so just to introduce myself for those who do not know me, I'm Krishna Balasubramaniam, and I'm a postdoctoral researcher at the University of California, Davis, recently assistant project scientist in the School of Vet Medicine. My research focuses on the evolution and behavior of non-human primates, specifically macaques, and the impact that social behavior has on health, of course, which is a very pertinent topic considering what everybody around the globe is currently undergoing. So basically, so my research is over the past 4 or 5 years in Davis focused on the influence of network connections on individual susceptibility versus the tendencies for animals to acquire and transmit infectious agents. And I've also of late been interested in looking at the impact of humans and anthropogenic factors on the evolution of behavior in non-human primates, and more broadly speaking, the impact of humans on the social behavior of wildlife. But today I'm not going to be talking about my research, but rather focus on like I said, a topic that's of broad interest, the implementation of social networks in behavioral ecology. And I'm going to be using some examples from across the literature so far on networks and its implications in behavior to basically demonstrate to you how networks have been useful in studying the evolution of social structure of wildlife, disease risk, and more recently, conservation. So without further ado, we can get going. So if you guys want to contact me about my research specifically, because I'm not going to be talking about that today, although there will be some elements of it that I will use as examples further down the line. You can always feel free to email me. You could also just look up my Google web page as well as my Google Scholar profile, and also follow me on Twitter. Okay, so this is the outline of the talk. It's going to be roughly 45 minutes. Basically, I'll start off by giving you a brief description of sociality and social structure, talk about why it's important to study social behavior. And then I'll get into a little bit of background on networks, specifically focusing on some of the terminology and measures associated with social networks, as pertaining to how they've been useful in animal behavioral research, specifically behavioral ecological research. The third part of the talk is going to be the longest part, where I'll be talking about the application of social networks in behavioral ecology, and I'll focus on certain subtopics more so than others, and we'll get there as and when we get to the third part. And I want to end with some cautions and considerations with regards to what researchers should keep in mind when they use networks to answer specific questions. So basically, what are some of the limitations of networks? What do you keep in mind while you use networks in statistical analysis and so on and so forth? So the fourth part will be the most boring part, but it's also the most important part, I should say. All right. So what is sociality? It basically is the tendency of animals to live in groups, and we can relate to that as humans because we are a group living species. So often at times, sociality and group living is of interest to a number of researchers. You're talking about anthropologists, behavioral ecologists, and of late epidemiologists as well, of course. And essentially, in animal societies, you think of group living as evolving when the benefits of living in groups, such as the cooperative rearing of offspring and predator defense, outweigh the costs, such as, for instance, the competition that animals face when it comes to accessing resources, as well as the susceptibility of individuals to acquiring and transmitting infectious disease. All right, so you see sociality as being ubiquitous across the animal kingdom. And you think of sociality, you think of three major aspects that are rather well studied. The first is social organization, which basically refers to once groups are formed, what do groups really look like? So essentially, you're thinking of factors like the sizes of groups, patterns of dispersal in terms of which animals move out of the group, which animals tend to stay in the same group, and also sex ratios in terms of the proportion of males and females in the group and so on and so forth. So essentially, what constitutes a group? Now, once groups are formed and once a basic social organization is sort of manifests itself, that kind of gives rise to two other aspects of sociality, which have more to do with behavior. The first is mating systems, which basically refers to the reproductive strategies adopted by males and females in the group. And you have a variety of different mating systems, even within the primate order, as pertinent to, of course, primatologists who constitute the majority of my audience today, I take it. But also the patterning and distribution of competition and cooperation in the group, which gives rise to what is called social structure. In other words, essentially, animals within a group may compete for resources, but also cooperate in order to access those resources. And the distribution of such behaviors within the group is often at times non-random. In other words, it's heterogeneous, but it has a certain patterning to it. And we'll see what I mean specifically with regards to that. And that's what gives rise to a group's social structure. Now, social structure is of a particular significance to us because it best captures the strategies adopted by animals to maximize the fitness of living in groups while minimizing the costs of group living. Okay, so essentially, I talked about two major aspects of social structure. One is, of course, competition. So I mentioned that animals often at times compete to gain access to resources, and the patterning and distribution of such competitive interactions culminates in what we call a dominance hierarchy. So essentially, you think of dominance as basically acts of aggression, typically, but you also have other forms of dominance. That's not important for the purposes of today's talk, but essentially, it's acts where an individual exerts its competitive or fighting ability over another individual. And such behaviors culminate in dominance hierarchies, which again are pretty ubiquitous across the animal kingdom. You see hierarchies from animals like ants and other social invertebrates to mammals that live in social groups, such as, for instance, wild canids like wolves. Okay. But on the other hand, you cannot have a functional group or a society where animals only compete. Often at times, the core of group living constitutes what are affiliative or cooperative relationships, because group living cannot manifest without animals cooperating to gain benefits. And one behavior, for instance, that is very commonly observed across the animal kingdom, particularly across mammals, is social or allo grooming, in which animals tend to clean or manipulate the fur of another individual. And often at times the patterning and distribution of grooming behavior, particularly in primates, is very heterogeneous. And that again gives rise to differences in their social structure. Now, the take home from this is that social behavior and particularly social structure may be thought of as being complex traits. So in other words, social groups are pretty complex systems. And this is where we really get into the realm of networks, because when you think of complex systems, you immediately think of social networks, as we segue into the second part of the talk, where you can think of networks as being patterns of associations of entities or nodes that are linked based on some shared characteristic. So basically, some form of interactions that these nodes experience. Now, you can think of biological networks at various levels of organization. So you can think of protein-protein networks at the molecular level, all the way up to ecosystems and food webs in which the nodes would constitute individual species, and then the links would be assigned based on the trophic level. In other words, the consumption of one species by another. So basically, you have networks being at the core of all complex systems, and especially biological systems. Okay, so now I want to get into some basic terminology when it comes to networks. So you think of a network, you think of nodes, as I just mentioned, which form the vertices of the networks. They're linked by some form of interactions that these nodes would share, which are referred to typically as edges or links. And finally, the whole system together is what constitutes a graph. So in network terminology, you can refer to the system as a graph or a network. And it's important to note that nodes and edges here can be anything, really. And you can think of networks that we expose ourselves to on a day-to-day basis, such as social media or Facebook in particular, where you have people on the web being linked based on their friends, but also not just their direct connections, but also who their friends are in turn connected to. And we'll get to talking a little bit more about that further down the line. So essentially, one network that all of us are a part of would be, or rather the good majority of people are a part of, would be social media. All right. Then here's another hypothetical network where the nodes represent actors in Hollywood, and the links are assigned based on the extent to which some actors act together in certain movies. And the nodes are also sized based on the popularity of the actors here. Obviously, from this, you can conclude that guys like Leo DiCaprio and Tom Hanks and somewhat controversially, I should say, Johnny Depp seem to be more popular than say some other actors who played roles in movies, but not as much as these big popular Hollywood stars. So essentially, this is a network of actors linked based on the shared characteristic being their tendencies to co-act in films. But let's move away from your day-to-day examples to biological examples, because that forms the core of this talk. So let's focus now specifically on ecological examples. You can think of networks as being bipartite in the sense that you basically have two major components that are not cross-linked to each other, but rather are interlinked across. So basically, you think of plants and their pollinators. You think of parasites and their hosts interlinked with each other. So these we typically refer to as bipartite networks. And then you have population networks in which the nodes represent a social group or a population, and the edges would be assigned based on, say, the migration or the immigration or immigration strategies of some individuals. So here you have an example from a study on vervet monkeys by Cheney and Seyfarth back in the 80s, in which each node kind of represents a social group sized according to the size of the group. And the arrows linking the group kind of represent movement of males from one group to the other. And you can see clearly that there is some heterogeneity there, in the sense that from some groups you have more males moving to some other groups, as indicated by thicker arrows. All right. And now we want to focus on social networks, which is going to constitute the crux of what we will talk about for the next 30 to 40 minutes or so, in which the nodes are essentially individuals in a social group, and the links are assigned based on a shared behavior or a characteristic that these individuals experience. So here in the center, you have a hypothetical network of, say, female primates, in which the alpha female is at the center of the group, and the arrows would be potentially grooming relationships that these animals experience. You can see that the alpha female tends to experience more grooming received than she actually gives, than say some of the other individuals who are middle to low ranking. As you can see, some animals both give and receive grooming, but in the case of the central alpha female in the group, she tends to receive more grooming. So essentially, this is a hypothetical network of grooming relationships in primates. But the links can be more than just one type of behavior. So the links can be assigned based on whether animals share a resource. As you can see in these ungulates in the top right corner of your screen, you see giraffe and zebra using the same water trough. And as a consequence, you can draw links between these animals based on the fact that they use the same resource. And then you can think of association patterns, like, say, animals just being in the same space, like, for instance, the zebra that you see in the bottom left of your screen. And then you can have non-contact social behavior, like, for instance, animals who are giving submissive signals to other animals. And finally, contact interactions like grooming, as you see in these rhesus macaques in the bottom right of your screen. So basically, the links can be anything. Even in a social network, you can have a pretty broad variation and heterogeneity depending on what the links are assigned based on. Okay, so I should say that over the past 10 to 15 years, network approaches have really revolutionized research in animal socioecology and behavior. There are a number of excellent reviews on this topic, and I want to point out a few that might be of interest to primate researchers in particular. The one by Cedric Sewer and colleagues here that got published in the American Journal of Primatology in 2011. That's a great read. And the book that I mentioned here on the right by Cross and colleagues is really useful in the sense that it partitions the use of networks chapter-wise, but also in accordance with the broad area of research interest. So for instance, it divides, say, reviews that focus on disease risk. There's another chapter that focuses on how networks are useful to study mating systems. And there's another chapter, for instance, on the diffusion of behavior and so on and so forth. So basically, it offers a more chapter-wise or a topic-wise review of the utility of social networks. Okay. All right. So let's talk a bit about some of the other characteristics of networks before we really go on into demonstrating how they've been used in behavioral ecology. We think of weighted versus unweighted social networks. And by unweighted, I basically mean you assign a link between animals based on whether or not they've experienced an interaction. So basically, the edges you see here in this network are all of the same thickness, and basically they're assigned based on whether or not the animals interacted in the first place. But then you can have a weighted network in which you actually take into consideration that some individuals interact more often or more frequently or for greater durations of time than say other individuals. So depending on that, you can change the weights, and as a consequence, you can account for heterogeneity or variation in interaction patterns. Okay. And then we talk about directed versus undirected. So basically you have edges that are assigned that may not have a direction. For instance, some undirected networks could be links assigned based on co-authorships in manuscripts, protein-protein interaction networks. But more pertinently, for the sake of behavioral ecology, you can think of networks where animals use the same space. In other words, do I share space with somebody else? And as a consequence, can I draw a link? Yes, I can. But then as to whether or not there's no the crux of it is that there is no concept of initiation or recipient or receiving in that case. Another example would be social huddling, in which animals just get together and huddle. In that case, there is no individual who's initiating the behavior and no individual who's specifically assigned to be the recipient. But then you have directed links on the web, directed links in other kinds of networks, such as, for instance, URLs on the web. So you can get from one web page to another. And so there's a clear sense of direction there. Metabolic pathways. And then in the behavioral realm, you have dominance interactions in which you have a clear initiator and a recipient. And you have grooming behavior in which you have one individual who grooms and then the other individual who actually receives grooming. Okay, and just talk a little bit now about how social network data are typically represented. So we use two approaches really. One is a matrix-based approach in which you have animals represented in rows and columns, and then you have interactions between those animals represented in the form of values in the cells. So basically here you have a hypothetical dominance network and an associated adjacency matrix in which values above the diagonal indicate wins from animals in the rows towards animals in the column. So basically here, individual 1 defeated individual 2 once, hypothetically. And in some cases, you might also have reversals. And what do I mean by that? Suppose individual 6 is rather low ranking. It does not necessarily mean that 6 would necessarily lose encounters almost all the time. It is conceivable that 6 might win the odd encounter. And as a consequence of that, you might have a link assigned in the bottom half of the matrix. So basically, this is a great way to look at network data. The other way to look at it is through using strings. In other words, you have instead of a matrix, you have animals represented in columns, in which you have initiators in the first column, recipient in the second, and then the frequency of interaction would be in the third column. Okay, so I want to talk now about some key network measures that can be calculated from such matrices and from such strings, which behavioral ecologists frequently use. And I'll get to some of the practical examples as we proceed into the talk. But essentially for now, what I want you guys to take home is the fact that that these measures are typically calculated either at the level of the individual, in other words, for each node, or at the level of the group. So you can have both individual level and group level measures. So let's talk a little bit now about individual level network measures. The simplest one would be degree, which basically refers to the direct connections of each node. So in other words, if I am linked to 4 other people in the network or 4 other individuals in the network, my degree would be 4. Then we get to slightly more complex measures, like for instance, betweenness, which basically asks the extent to which a node would lie in between or in the path of other network node components. So basically, you think of these 2 nodes here that I've circled. You think of nodes that are high in betweenness because they essentially act as links to major components of the network. So it basically asks the number of links that pass through the node that also connect other nodes within the network. Right? And then you think of eigenvector as not just the number of direct connections that a node has, but also how well connected a node is to nodes that are also well connected. So what do I mean by this? You think of these nodes here, and you think of their direct connections. They're essentially just connected to one other node, and that would mean their degree is pretty low. So they'd have a degree of 1. But then eigenvector is more than just your direct connections. In other words, it asks whether you're connected to nodes which are also really well connected. So basically, if you were to consider secondary and tertiary connections, these two nodes would actually be more central in the network than you would expect if you were to consider only their direct connections. And you'll see soon enough as to why secondary connections matter and how they're relevant to behavioral ecological research. Okay, so I mentioned that there are some measures that we calculate at the level of the individual, but others that we focus on that are calculated at the level of the group. So you talk now about I just talked about centrality. And if you extrapolate variation in centrality to the level of the group, you can have networks in which some nodes are more well connected than others. So basically, what do I mean by that? You look at this network here. It shows some nodes that are sized larger than others. And that's based on the fact that they have more direct and secondary connections than other nodes. So that can indicate the skew or the difference in centrality, or the extent to which at the group level you find a skew in centrality. In other words, the extent to which some animals seem to be more important or seem to be more well connected in the group than, say, other animals. Okay, so here are 3 hypothetical networks, and these kind of demonstrate really well as to what the centrality coefficient gets at. The network that's labeled A basically has every node connected to practically every other node in the group. So basically that has a centrality coefficient of almost 0, because all of the individuals are equally and extremely well connected. But on the other hand, network C represents a case where you have one individual at the center who's connected to several others, but then these other individuals who are not connected to each other. So basically this is a star network. In other words, one animal is highly central, but then all of the other animals are quite peripheral. So basically, in that case, you have a very high skew in centrality. Okay, and then you think of other measures like density, which is basically the number of edges over the total number of possible edges. So you can have some networks that are more densely connected, like the one on the left, than others which are more sparsely connected, like the one on the right. And these refer to examples from data on macaques that I'd studied for part of my PhD work, and we'll get to more about differences in social networks and macaques as we proceed with the talk. Okay, and then a third measure would be modularity or substructuring. So this basically refers to the extent to which a network forms substructures or subgroups. So basically you have certain cases where you might have a tendency for some individuals in the network to interact more among themselves than they do with some other individuals. And here you have an example of a network of rhesus monkeys connected based on their grooming relationships. Similarly colored nodes basically refer to individuals that are part of what we call the same cluster, because they interact more among themselves than they do with individuals in another cluster. And so there are various algorithms that are available to calculate community substructuring or modularity that are beyond the scope of this talk. But if anybody's interested, the source papers are down below. Essentially what they do is they assign or pre-assign communities of various sizes, and they calculate some function of the relative proportion of edges that are within the assigned communities compared to the proportion of edges that go across these communities. And they do this iteratively for different sizes of communities until they reach a most probable community membership. Okay. So you have other ways of visualizing modularity, which we refer to as tree building or clustering methods. And two examples would be hierarchical clustering or data cloud geometry. What these methods do, they typically consider interactions among individuals in a group, and they cluster individuals that tend to interact more similarly in the same cluster. And they do so in a manner such that similar or identically interacting individuals would be at a shorter distance in terms of this tree structure from communities that are more distantly interacting. In other words, you have both community membership assigned as well as the distance between communities defined in terms of the extent to which there is inter versus intra-community interactions. Okay, so with that introduction, I just want to proceed to the next part of the talk, which basically will focus on how these network measures have been used in behavioral ecology, more broadly speaking. I just talked to you about a number of measures at the individual level and at the group level. But then what is all this in aid of? Why is modularity important? Why is it important to know whether or not some individuals are more central compared to others? So that's going to be the crux of what I'll be talking about for the next 20 minutes or so. And I'm going to be focusing on certain topics more so than others. So I should point out before we get into the details that social networks have found broad applications in behavioral ecology, particularly over the last 15 years or so. And there are 5 major areas that I've outlined here, and there may be more. So you think of social structure and basically defining and comparing the social systems of animals. You think of disease risk. In other words, networks have proved useful tools to model the acquisition and transmission of infectious agents. And that's pretty pertinent to what everybody is undergoing at this point in time. Conservation, in other words, the impact of humans on animal space use and social fragmentation. So networks have started finding use there. And then finally, a couple of topics that I'm less familiar with. In other words, the diffusion of information or the spread of novel behaviors through a group and collective movements. In other words, whether in animal groups there are specific leaders and followers. And if so, whether social networks determine the extent to which some animals tend to decide movement patterns and are followed by other individuals. So basically, I'll focus less on the last 2 topics. There are some really cool studies, particularly with relation to the diffusion of information on humpback whales and chimpanzees, which I can talk to you guys about much later. But then for the purposes of this talk and given time constraints, I'm going to be focusing on the first 3, and the approach is going to be this. So I'm going to be introducing each of these areas and then sort of going over a couple of case studies in each area that are pretty pertinent to examining how networks have found use in these areas. Okay, so first let's look at social structure and just describing and comparing animal social systems. And this is of particular interest to primatologists because non-human primates show a broad variation in their social organizations. So you have solitary species like mouse lemurs, all the way up to fission-fusion societies that chimpanzees typically show, in which you have constant changes in group composition and community composition in particular. So you have a broad variety of social organizations. And as you can imagine, because I just mentioned earlier that social organization underlies variation in social structure, if you're going to be finding variation in social organization, it's pretty much nailed on that you're going to be finding broad within and between species differences in social behavior and social structure. So now I'm going to be talking to you about a couple of case studies in which networks have have been used to describe the social behavior or social structure of group living animals. The first is a study on harvester ants, which I'm particularly fond of, which was conducted in an experimental setting by Noah Pinto-Wallman, who was at UC Davis before I came to Davis. The study is really cool for many reasons, but I'm going to really be focusing on on their findings with regards to the variation in centrality distribution or skew. In other words, basically what they did was they had these experimental settings in which the nests of ants were kept within these nest chambers, which were connected to tubes which allowed dispersers to leave the nest and return to the nest after foraging. And then they monitored the frequency of interactions between foraging ants as they entered the chamber, both in terms of where the interactions occurred, as well as with whom interactions occurred more frequently. So what they found was that there was a broad variation in network centrality, and that variation was essentially explained by the path length of individuals once they left the nest. In other words, the longer or the more convoluted the path that animals traversed, the more likely they were to be more attractive partners when they returned to the nest. So you can see the network graphs here on the left, in which the nodes are colored based on the centrality. And you can see some individuals are highly central as compared to others, which are more peripheral. So essentially what they showed was that there was a skew in centrality. We just talked about centrality skew a few minutes ago. Essentially, we talked about societies where some individuals end up being more important than others, just because of the fact that they carry information or they have greater access to a resource. In this case, it is the former, in the sense that ants that had more convoluted path lengths were presumed to carry more information, and as a consequence, you had a skew in centrality with regards to with regards to some individuals being more important than others. And they also used what was really cool was that in addition to showing this difference, they also used mathematical models to demonstrate that this gradient in centrality actually facilitates the flow of information through a network. In other words, it actually accelerated the speed with which a potentially novel piece of information might spread through the social group. Okay, and now we get to examples at the group level, and to I guess a number of people in this audience would be familiar with macaques and the variation in social behavior that they show. It is also pertinent that I talk about macaques now because apparently it's International Macaque Week. What's more, macaques have been the focus of my research for the past 8 years or so. Essentially, they're a fascinating genus to look at variation in social structure and social networks, because a number of these primates show broad similarities in social organization. In other words, they all tend to live in groups in which females form the core of the group, or females are philopatric, and males tend to disperse between groups. But at the same time, they also show marked variation in social behavior. By variation, I'm not just talking about the frequency of behavior, but also the distribution of behaviors like dominance and grooming. And that's led a lot of researchers to actually classify macaques into social styles. Now, the social style idea is somewhat controversial, but the crux of it is that macaque groups and species may fall under a continuum that range from despotic species that typically show steep dominance hierarchies, low levels of post-conflict affiliation, and pretty intense degrees of preference towards kin or kin bias. So some examples would be rhesus macaques. But then at the other end, you have what are more egalitarian or tolerant social systems that you see in some of the Sulawesi macaques of Indonesia. Now, these species tend to show more relaxed dominance hierarchies or shallow hierarchies, higher tendencies to reconcile after conflict, and also lower degrees of kin bias or preference towards kin when it comes to affiliation. And so a lot of researchers look to see whether variation in social networks sort of reflected this variation in social style. And it so happened that some research has suggested that at one end of the scale, you had macaques that showed modular centralized networks. In other words, you have despotic species in which you also have preference of animals of grooming close kin, showing modular grooming networks. In other words, networks in which you had a greater tendency of individuals to groom closely related individuals compared to more distantly related individuals. Okay. And then at the other end of the scale, you have tolerant species or egalitarian species, which showed more dense or decentralized networks with lower kin bias. In other words, you had individuals interacting preferentially with both kin and non-kin, and as a consequence, you had more denser, more well-connected social networks. So there was a broad difference between species at the two extreme ends of the scale. And what's more, evolutionary biologists and anthropologists have linked such differences to both ancestral characteristics, like, for instance, species phylogenetic relatedness, as well as to adaptation by animals to variation in current conditions, like group size and variation in resource abundance, and so on and so forth. So basically, you have broad differences in social networks across this genus, and networks have helped capture the differences in social styles of macaques. Okay, so now I'm going to be talking a little bit about an area that's particularly prominent in the current climate that we're all experiencing. In other words, the application of social networks in behavioral ecology as pertaining to infectious disease acquisition and transmission. Okay, so social life has a profound impact on health, and I need not elaborate on this because I'm pretty sure that almost every person on the planet at this point in time is experiencing this. So with regards to disease risk and behavioral ecology and wildlife ecology more broadly speaking, there are a number of reviews of social networks that have focused on how animals that are well connected in their network might also aid in the acquisition, or I should not say aid, I guess, may also function in the acquisition and transmission of infectious agents. So over the past decade or so, you have a number of reviews on this topic that have intensely focused on how networks may act as a scaffold, if you will, for the transmission of pathogens. So some of the common frameworks or hypotheses that are focused on this with regards to animal behavior would be the contact transmission or super spreader hypothesis, in which animals that are highly central in a network might be more prone to acquiring and transmitting infectious agents. But then on the other hand, you have the social buffering hypothesis, which suggests that central individuals, also in lieu of having enhanced connections or strong social bonds in some societies, might experience greater social immunity and as a consequence be more resistant rather than be more susceptible to acquiring certain types of infectious agents. So you have these contrasting hypotheses at play, which I should point out are not necessarily mutually exclusive. Okay, and then finally you have the social bottleneck hypothesis, which suggests that community structure, and we talked a little bit about this when I talked about modularity. Community structure leads to the trapping or the social bottlenecking of infectious agents to the point where you would expect infectious agents to spread faster within communities compared to, say, across communities. Okay, so I'm going to be reviewing some studies on each of these three hypotheses. So first, let's look at contact transmission. And there was a study on Belding's ground squirrels conducted by some researchers in my lab before my time that focused on juvenile males in these ground squirrels and asked whether males that connected burrows of ground squirrels were more likely to be super spreaders. So basically, what they did was they reconstructed networks based on connectedness of animals, and then sort of evaluated the prevalence of a protozoan parasite, Cryptosporidium, in these ground squirrels, and essentially showed that central individuals had a higher risk of infection. In other words, you can see here this graph on the left, which has black nodes that are colored based on individuals that had infection or had Cryptosporidium, compared to white nodes where individuals were not infected. And as a consequence, they showed a link between centrality in the network and the likelihood of being infected by a protozoan parasite. But the other interesting finding from this was that there were some individuals which, because they clustered more among a subset of animals, showed a lower prevalence of Cryptosporidium. In other words, what do I mean by clustering coefficient? It refers to the extent to which there is a node that connects a subset of other nodes in the network and forms these triangular triplets. So basically, the idea is that the more clustered you are, the more likely an infectious agent may be trapped. And as a consequence, you might reach a time where the pathogen survival time sort of diminishes. And as a consequence, you might expect to see lower prevalence of pathogens in more clustered clustered networks than say in non-clustered networks. So this was a pretty interesting study that focused on individuals in a network and contact-based transmission. Okay, and then you have the social buffering idea, which I'm going to be illustrating using a study that I did on captive rhesus primates with a lot of my colleagues here at UC Davis at the California National Primate Center. Essentially, we looked at social networks of rhesus macaques, and we asked whether centrality in the network, as indicated by eigenvector, was linked to the acquisition of an environmental pathogen, Shigella flexneri. And as the graph on the right shows, we found that animals that were more peripheral in the network, or rather the nodes that are smaller in size, were more likely to be infected by Shigella than nodes that are more central in the network. So in other words, here you have evidence for social buffering in the sense that animals that were more well connected, in other words, not just connected directly, but also connected to animals who were in turn really well connected. In other words, individuals which had high grooming eigenvector were the least likely to be infected, or to flip that around, animals with the lowest grooming eigenvector centrality and it so happened huddling degree were also the most likely to be infected. Okay, and third, let's talk a little bit about community membership and how communities may function as social bottlenecks for the acquisition and transmission of infectious agents. And for this, I want to illustrate a study done by Charles Nunn and colleagues that used agent-based models to look at transmission of infectious agents or artificial infectious agents through artificially constructed social networks. So basically, for now, you don't need to worry about these complicated equations in the bottom. But the crux of what you need to take home from this is that agent-based models essentially divide agents or artificial agents within a group into three categories: susceptible, infectious, and recovered. So essentially, this is the simplest agent-based model, or it's called the SIR model. Now, what researchers typically do is they introduce an artificial infectious agent into a social group and model iteratively the extent to which it is transmitted across the group. So the transmissibility can be modeled in terms of both individuals' connections. In other words, I, as you see here, is it represents how well the first infected animal is connected to other other individuals who are susceptible, and then beta sort of represents the per contact transmission probability of the pathogen. So basically, it models infectious agent transmission based on assigning agents into categories and then iteratively modeling the spread of infectious agents. So what they did was they caused network connections as well as host and pathogen characteristics, such as, for instance, these values here, beta and I, to vary stochastically, and also some transmission parameters. And they sort of set certain limits and constraints for within versus between subgroup infection probability. And using a number of simulated networks, in other words, 1,000 randomly generated networks, they model the transmission dynamics of infectious agents. And when they did this, they ran models until such time that either the pathogen completely died out, in other words, the recovery rate of individuals exceeded that of the infection rate, or they died out until they ran the simulations until a saturation point was reached. In other words, the maximum possible number of individuals that were infected, that could be infected, ended up being infected. And that is basically what this value of R indicates, or R infinity. It is basically so basically it's an indicator of the number of infected individuals at the end of the iteration. Okay, so when they did this, they basically plotted the prevalence of these infectious agents in networks where there were subgroups, and compared them with networks where there were no subgroups present. And when all other parameters were kept consistent, they found that subgrouping made a big difference. So really the take home is that when there were subgroups present in the network, the overall prevalence of the infectious agent at the end of the simulation run was much lower than the prevalence of the infectious agents in cases where there were no subgroups present. And they did this for a variety of different infectious agents with potentially different rates of transmission. And so this is a pretty comprehensive analysis that covered a broad suite, if you will, of pathogens and transmissibility rates. Okay. So next, I just want to talk a little bit about some studies that have started using the phylogenetic relatedness of gut microbes to model transmission dynamics. And essentially what these do is they conduct microbial sampling in wildlife populations or animal social groups at specific time points. They watch animal social groups to record behavior over a particular period of time. And essentially they compare the genotypic or microbiome similarity of individuals across a particular time point during which behaviorally they were sampled, and ask whether there are links between social behavioral networks and say microbial phylogenies or microbial transmission networks. And here's a really cool study on ungulates, again, done by some researchers in my lab before my time at UC Davis, that looks at the comparison of social networks of ungulates, specifically giraffe in Kenya, and compares networks of individuals based on their association patterns with networks that were constructed using the similarity of gut microbes, specifically gut E. coli. And here you can see a strong correlation between animals' association strength and the probability of sharing the same subtypes of E. coli. And I sort of extended this to look at rhesus macaques at the primate center here in California. And my research here has suggested that when you look at the community level, in other words, when you look at communities of closely interacting macaques, they tend to show greater similarity in their genotypic profiles of E. coli than macaques that are formed across communities. So basically, the plot here on the right shows you the percentage of similarity in E. coli of macaques within the same community, which is indicated by the green box and error plots, compared to macaques across communities. And you can see that in 3 groups here, we found evidence to suggest that there might be social bottlenecking. In other words, greater similarity of E. coli among individuals within the same community. Okay, so just to wrap up this whole disease transmission subsection, these are just we're basically scratching the surface here when it comes to networks and their utility in studying disease risk in animals. Disease transmission is temporary and may depend very much not just on the networks of the hosts, but also on the characteristics of pathogens. In other words, their transmissibility. And so you can have a case where you might be able to identify potential transmission dynamics if you do more temporal sampling. In other words, not just look at one time point, but look to sample as frequently as possible in order to detect these transmission events based on contact behavioral patterns. Okay. And as I just mentioned, these also depend on pathogen-specific characteristics, such as the incubation time and the contagion period of specific microbes over others. That needs to be factored into a number of these models. And also, when it comes to targeting super spreaders for vaccination, this is a really useful tool in the sense that when we have information related to animals' behaviors, and specifically their positions within social networks, it might lead to more invasive approaches to identify potential target individuals to control the spread of infectious disease. Okay, so the last part of this, I'm going to be very quickly focusing on what is a new and developing area of research where networks have found use. In other words, the impact of humans on animal space use and social fragmentation, which of course would have strong implications for conservation. Okay, so human-wildlife conflict doesn't really need introduction to audience, and essentially you can think of a number of examples where humans have impacted wildlife negatively. You can think of landscape changes. You can think of the culling and predation of animals, poaching, translocation of potentially problematic animals, noise pollution, and so on and so forth. The list is pretty endless, but essentially, this might impact wildlife in different ways. And some of the more visible ways would be when it comes to impacting the mortality of animals, changes in population dynamics, but also more subtle impact would be changes in their behavior or social behavior. So this whole subfield of research when it comes to studying social behavior from the point of view of conservation has again taken off this past couple of decades, but only recently has social networks really come to the fore with regards to conservation. And I want to just review certain ways through which networks might be useful in the context of conservation. And I'm going to be borrowing a lot of this from a review by Schneiders et al. In 2016 that focuses on how networks can find use in wildlife conservation. I should point out that there are not too many empirical studies that have come out in this regard, and that's also because this is a pretty emerging area of work. So rather than actually point you guys to specific examples, I'm going to be just focusing on giving you an overview. So you can think of one way through which it might impact would be the impact of landscape changes on social network structure. So basically, you can think of humans impacting a forest by removing valuable resources. And as a consequence, resources might become more widespread or more clumped. And that can change the network of, say, spatial associations or social interactions of group living animals, such as, say, elephants. And you can have more fragmented networks as a consequence, where you might expect greater modularity, greater connectedness or fragmentation. I should say lower connectedness or greater fragmentation, lower density, And also you might see both a decrease in aggression because of the fact that animals very rarely come into contact with each other, but also an increase in aggression because now resources are all of a sudden more clumped. And as a consequence, there is more competition for these resources among subsets of individuals. So you might have very localized aggression that's artificially inflated by the fact that humans have had an impact on resource abundance and distribution. Okay. Another way would be the removal of some individuals, and the removal can happen because animals are often at times trapped and relocated because they're considered to be problematic. And this is often done in a very haphazard way without conservationists, or I should say, without policymakers realizing the implications that might have for the stability of their social networks. So you can think of losses of specific individuals as having a major impact on network structure and the long-term resilience of networks. So you remove all of a sudden a couple of individuals that were central to this network here. You see a number of links lost as a consequence of that. And you might have a situation where the population then needs to be monitored on a more longer time scale in order to detect or see whether animals readjust their strategies. In other words, do they form fresh relationships or fresh connections as a consequence of losing connections? And that would give you information with regards to how animal social networks might change. You might then expect to see a greater increase in fragmentation or modularity. And then all of a sudden there might be a decrease because there is greater social rewiring and so on and so forth. So it's really vital to track what I call temporal changes or variation across time in social networks after humans have had an impact on these networks. And finally, I touched a bit upon this in the disease transmission section. Another key application of networks with regards to wildlife conservation would be the selection of some individuals to vaccinate or potentially treat to effectively block the acquisition and transmission of infectious agents. So basically, let's say you have a zoonotic agent entering a population. You might, if you have prior information on the network structure or the behavior of this population, you might already have information with regards to certain individuals who might perform or because of the fact that they are more central in a network might tend to act as super spreader of infectious agents. And so it might inform conservationists with regards to the strategies they implement to trap certain individuals to treat them before they're re-released into the wild. So basically, having prior knowledge on social behavior might be vital to implement invasive strategies with regards to mitigating zoonotic disease transmission. Okay, so with that, I'm just going to wrap up very quickly. I already see we've gone for an hour now. So I'm going to be wrapping up with some cautions and considerations while using networks in statistical analyses, which a number of you, particularly in the AIP group, might be doing or might be wanting to do in the near future. So I just want to touch upon a couple of things to keep in mind. Okay, so one thing to note is that when you use networks in social behavior and statistical analysis, it's important to note that they are susceptible to sampling effort. In other words, you have to ask the question as to whether you have an adequate sample in order to construct meaningful networks. So the first approach to do that would be to compare observed values to those from random graphs. So in other words, what you do is you generate, keeping certain aspects of the network the same, for instance, say the number of nodes or say the total number of edges, depending on your question. You can keep certain aspects consistent, but basically use that to generate a number of random networks and ask whether your observed network values are significantly different from random. Another approach would be to plot networks as a function of cumulative observation time. So in other words, you divide your data set based on some temporal time period through which you have sampled your group, and then plot calculations of network measures as a function of time. So you ask whether after a particular point in time, your network measure basically stabilizes. And as a consequence, you might be able to conclude that you have adequate amount of data. So I should point out here that neither of these approaches are completely foolproof. They both have their strengths and weaknesses. It's just important to keep in mind that this is a very hotly debated topic. Even as I speak, there's a lot of contention with regards to what the best approaches might be with regards to using networks in statistics, more broadly speaking, but also with regards to figuring out if you have an adequate sample size. Okay, so speaking of using networks in stats, many of you might run linear models to look at how, say, certain predictors, such as, for instance, the size of your group, or say some attributes of individuals like age or sex or dominance rank or so on and so forth, might impact their network position. So basically here, network positions would be the outcome variables. It's important to note that when that is the case, you're dealing with a set of measures that are not independent. So they are highly interdependent in the sense that you're calculating these measures based on links, not just assigned to one node, but also invariably assigned to nodes that are also well connected to other nodes. So basically, there's a lot of interdependency in the data, which you would need to account for. And one way to do that, and again, this is not a foolproof way, but one way that researchers have popularly done this would be to use permutation-based models in which you basically swap the nodes in the network that you have and generate, again, random graphs or random data sets by either swapping the nodes or swapping the edges, and end up comparing your observed regression coefficient to, say, a distribution of coefficients obtained from permuted data or random graphs. So this is a great example that Damian Feirin illustrates in his review of social networks in 2015, in which basically the effect of sex on the strength of social networks is examined. And when you run the model, it shows a difference in the sense that males show markedly more network connections than females. And although the regression model doesn't pick this as being significantly different, when he plots, say, the coefficient against, say, a set of permuted data or the coefficients generated from permuted data, he does find that there is a significant difference. So essentially, here you might be validating what is in fact a clear result by using permuted data sets. Okay, so there are a couple of ways to do this. First, you could do, like I said, edge swapping or pre-network randomizations. The other approach would be to do post-network randomizations or node swapping. And it's been shown to be the case that doing it the first way, or rather pre-network randomizations, allows you to avoid sampling bias in the sense that this is another example from a follow-up paper by Feirin in 2017, in which he basically illustrates the usefulness of these permutation-based approaches to avoid type 2 errors. In other words, type 2 errors, as you might know, would be basically accepting a null hypothesis when in fact you should be rejecting it because you actually have a significant result. The plot on the left shows you the difference between females and males in terms of the strength of the social networks. You see clearly that females showed a stronger relationship, or rather a more strongly connected, or I should rephrase that. So basically, females were more strongly connected in their social networks than males, and that's evidenced by the plot on the left. But then they basically removed 20% of the data from females, and that result was lost in the sense that the plot on the right, all of a sudden, when the data is removed, showed no difference between females and males. That they attributed to the fact that there might be, in some real life situations, a sampling bias where just because males, say in say societies of birds, males are more brightly colored, and so you might have a tendency to excessively sample males more so than females, and such a sampling bias might lead to you not being able to detect what is a clear difference between female and male network connections, just because of the fact that you have less data on females compared to compared to males. And they illustrate that using a pre-network randomization, you can get around that, because what you're doing is essentially swapping edges and generating random data, and you're comparing a coefficient based on these random data to the coefficient that you observed based on your data set. So when they used the pre-network randomization, they did find that they could avoid this sort of error that might come across as a consequence of sampling bias. Okay, so essentially what this does is show that although you might have inter-individual variation in observation time, in other words, you might have a case where you, in your study system, you might observe some animals more often or more frequently than, say, others, you could avoid a type 2 error in such cases by resorting to using random graphs. Okay, but just to on a pretty objective note, I just want to point out that networks should be considered to be a tool by themselves and not something that you would potentially use under all circumstances and scenarios. So essentially, you should come up with hypothesis-driven reasons to use networks in the first place. And then as a consequence of that, sort of work your way into deciding what nodes, edges, and measures would most suit the hypothesis that you're trying to test. In other words, don't think of throwing the kitchen sink. You might be tempted to measure everything under the sun. You might think that modularity is really cool. You might think that centrality in terms of direct or secondary connections might be awesome. And just because of the fact that you might be captivated by some measures more so than others, there is a temptation to calculate everything under the sun and basically just ask if there are differences in one or more of those characteristics. I would strongly recommend avoiding doing that and basically adopt a very data-driven, hypothesis-driven approach to deciding how and whether you would use network measures in the first place. And I want to point to a recent paper that came out just a few months ago that really gets at this in a cool manner. Because Sebastian Souza and colleagues, they use what are essentially decision trees which guide researchers into making informed choices about what network measures are more suitable for their purposes. So basically, I strongly recommend that you guys take a look at this paper before you decide what measures work best for you, given the research question that you have in mind and the study system that you're focusing on. All right. So with that, I just want to end the talk. So I'm going to be stopping here, and I'll be happy to take any questions.
Okay, so thank you, Krishna. That was a wonderful talk.
Just a minute. Yeah, go ahead.
Yeah, so yeah, that was a wonderful talk. Thank you. I'm sure all of us enjoyed it. So there are a few questions and there are a few compliments, I would say. So Kevin Rosenfeld says hi and compliments you for this talk. And also, Rahim says that it's an excellent presentation and all the slides were like clearly visible.
So just give me 1 second. Just give me 1 second. I'm gonna.
Okay. In the meantime, there were some compliments for the initiative that AIP has taken. So I'm just going to read those compliments. So yeah, Rishi Kumar compliments AIP on this initiative and he hopes that the AIP continues to organize such talks. And also Shraman Das Gupta has complimented AIP on this initiative. So Krishna, are you ready to take questions?
Yeah.
Okay, cool. So first question is by Dipanvita Purohit. She asks, how do you address the observer bias while designing a social network of animals of larger group size?
So that kind of alludes to the last couple of slides that I was putting up. So in larger groups, you would potentially need to cater data collection such that you get adequate information, of course, on all the individuals to give you a more informed network in terms of not just having data on a subset of individuals, but also but more so on the majority of individuals possible. But in terms of observer bias more generally, I mean, it would help to know what exactly where exactly the observer bias is coming from, in the sense that if you are more prone to collecting data on some individuals more so than others, I pointed out to random graphs and using edge swapping as a way to look at whether your difference when it comes to calculating a network measure might be because of a difference in sample size, or rather in a difference in observation time versus a real effect of say a biological effect that you're seeing. So basically, one way to get around it would be to use, like I said, random graphs. But then larger groups, I should say more generally, present a challenge because it is difficult to collect adequate data uh, on all the individuals in the group for sustained periods of time. So you can get to the stage of using random graphs only after you have a threshold amount of data already on the individuals, and so that in itself would present a challenge when it comes to larger groups. You might resort to sampling approaches that would particularly in certain restricted settings, like say captivity, or in semi-free ranging settings, you might choose to observe say certain parts of the home range of groups, and use what are called event sampling methods, where you divide the home range into specific sub blocks. And so you go to a block for say a period of time and watch and record all interactions that might take place within that block. And so as a consequence, you also generate what are essentially non-independent data points. But then this is again clutching at straws, in the sense that, like I said, larger groups always present a challenge, more so than smaller groups. And so only after a particular threshold amount of data is collected can you actually even resort to looking at random graphs to see whether the effects that you're seeing might be a consequence of inadequate sampling versus true biological effects.
Yeah. So the next question is by Arijit. So he asked, like, how can we use SNA to model human-animal interaction and potential risk of transmission of zoonotic diseases in a human-wildlife interface?
Yeah, it's a great question. So that's one thing that I didn't really touch on now, because basically not much research has been done. So this was a review. This was an overview talk, essentially, of what research has already been done. But I have given this question some thought, as have some other researchers. And I think there are a couple of different ways to do it. One, of course, would be to actually have information on humans and include humans as nodes in what would be essentially a multimodal network. So, but what do I mean by multimodal? It would basically assign links not just based on, say, some subsets of individuals which constitute a group, but also include individuals from, say, other groups. So you're basically assigning links both within a group and across groups. And in this case, the other group would be, of course, humans. And in that case, you might have a situation where ideally you want the identities of individuals at the interface who actually interact with your animals, but that's not always possible or feasible. So what researchers might think of doing instead would be to look at broad demographic characteristics of humans. Like say, visitors versus tourists, or say age, sex classes of people who interact, like males versus females, and so on and so forth, and draw links based on animals in shared interactions with humans of specific demographic characteristics. And when you do that, it's possible to not just use these multimodal networks to model transmission, but also from these multimodal networks, you could construct essentially what is a social network. In other words, you can link animals based on the similarity with which they interact with people. So in other words, when two animals interact with a human at the same time, or consecutively within, say, a particular time period during which you expect infectious agents to be transmitted, then you might be likely to draw a link between those two individuals based on the fact that they shared a similar spatio-temporally similar interaction with humans. So there are a couple of different ways to do it, which researchers are exploring. To be honest, I'm not aware of any research that has actually gone ahead and constructed human primate interaction networks, or rather human wildlife interaction networks, and modeled disease transmission. But if somebody has, I'd be delighted to take a look. Yeah.
Cool. So there's another question by Saili Sawant, and she asked that, can we do a social network analysis in groups where only few individuals are identified, or no individuals are identified?
So that's going to be difficult, if not impossible, I would say, because you need to know the identities of the individuals. Because basically, when you think of social networks, you think of nodes as being individuals and links being assigned based on some shared characteristic or shared interaction between these individuals. And as a consequence, one of the main reasons to reconstruct networks and use networks in animal behavioral research is to look at variation in contact patterns between individuals, and as a consequence, look at how group structure sort of emerges from that variation. So unless you know the identities of the animals, it's going to be complicated.
Yeah. Okay, so there are just two more questions. I'll just quickly go over it. So next question is by Sayan. He asks if you can provide leads on using social network analysis in understanding cross-species specialities where one of the species is human.
Okay, so this again, I think this is very similar to the question, the earlier question, which is, are there any examples of human wildlife interaction networks? And essentially my answer is the same, which is that there's not much research that's been done, although researchers have started speculating on how best to do this. And there are a couple of different ways to look into it, as I just mentioned. One would be to assign humans as nodes, and the nodes would be described based on some broad demographic characteristics, like say visitors versus tourists, or say the age, sex categories of people who interact with wildlife, but also link animals based on the similarity with which they interact with humans. So there are, like I said, there are a couple of different ways to do this, but nobody's, to my knowledge, it's not been, it's certainly from the point of view of analytical approaches, we are only scratching the surface at this point. Yeah.
Okay. So there's this last question by Randeep Singh. So he wants to know if the social network analysis can be done in a solitary carnivore, and if you have any suggestions for that.
So that would really depend on the question at hand. Solitary species, researchers do use social network analyses, but not necessarily to describe their social systems, because obviously you're talking about solitary species. But I can think of examples where you might link solitary carnivores, for instance, based on the degree to which their home ranges might overlap. For instance, if you're thinking of, say, a population of tigers in Mudumalai and Bandipur combined, you might ask to the extent to which the home ranges overlap, and as a consequence, each animal can be a node, and the edges can be assigned based on the extent to which they share home ranges. So that's one example I can think of. But then, like I said, it would really come down to the research question that you have. So I would not really bother myself with constructing such networks unless you have a clear question. Are you actually really interested in looking at home range overlap? What is the usefulness of doing so? How is it going to ultimately aid your research? What is the end goal here? So you have to ask yourself those questions before you even consider networks, never mind actually implement them. And it's pretty easy to get carried away by social network analysis to the point where you want to just use networks everywhere and across circumstances, and that's not possible, and that's also not how science is typically
advisable.
done.
Yeah,
It has to be hypothesis driven, and it has to be informed based on the question, the larger question that you want to address. Yeah.
Cool. Hey, so Krishna, like there are a few questions. So would you mind sticking around and answering them, or?
Sure. Yeah, I can stick around for a few minutes. Yeah.
Okay, cool. So there's a question by Anupama Devi. She asks if is it possible to study evolution of host pathogen interactions using network analysis?
Yeah. So I can think of essentially I touched upon the idea of bipartite networks during my talk, which basically link two categories of nodes across each other, but not necessarily within the same category. So I gave the example of plant pollinator networks. And yes, there has been research that has been done that has focused on parasite host interactions in a similar way, or pathogen host interactions using bipartite networks. Again, it's very early stages, but I'd like to allude to one study that for me was super interesting. It came out in PNAS in 2013 by Gomez and colleagues, and Charles Nunn was the corresponding author, in which basically they linked bipartite networks of primates based on primate species that share that basically were infected by parasites. So basically, they drew links between various species of primates and the parasites that typically infected them. And then they used these bipartite networks to almost project into what was a social network. So basically, they linked first they linked primates and their parasites, and then they linked primate species based on their number and the extent to which they shared parasites. The purpose of doing that was to ask whether some species might be more prone to be potentially influential in emerging infectious disease research. And if you really want to get into host pathogen interactions from that perspective, I suggest that you start by looking at that paper, because they use phylogenetic approaches and least squares regression approaches to particularly show how certain primate species, just because they're more well connected to other species in terms of sharing parasites with them, might be the foci of research down the line in terms of recognizing and ultimately stopping emerging infections.
Okay, cool. So the next question is by Dolly Bora. She asks if any measures that can be considered for low sample size while doing social network analysis. Are there any measures that can be considered for low sample size while doing social network analysis?
So that would again depend on the question more so than anything else. So when you think of low sample size, you have to weigh up essentially the sample size that you have with the question that you want addressed. So I would make decisions more based on the research question that you have first, and then focus on the sample size and ask whether your sample size would allow you to address that question. Now, with regards to low sample size and networks, it's pretty difficult to sort of draw conclusions based on that, unless you more specifically define what is low, and how low your sample size is. So it's all relative. And the general idea is that the greater the sample size that you have, the more robust your network measures are likely to be. But that also would reach a particular threshold. So it's the question of sample size and its impact on networks is pretty complex, in the sense that it's difficult to answer unless you sort of or rather I should say until you consider your study system and your research question as well, while making that decision.
Okay. So there are just two last questions. After that, I think we're not going to take more questions. She's
Yeah. Yes.
curious cat.
Yeah.
Yeah. So there's just two last questions. The next question is by Shraman Dasgupta, and he asked, like, if a particular social behavior of a species restricted in its distribution is due to something specific to its range, how to determine it to be so? Like, had range been more, would the behavior be different?
So basically, you're the to sort of let me see if I'm understanding this right. He's interested in the impact of home range size on variation in social networks. Is that
Yeah,
essentially it?
that's the gist of it. Yeah.
Yeah. So basically, you think of animals that use larger home ranges. You also think of groups that typically are larger in size and also might be more spatially widespread. So as a consequence, you might find more clustered social networks in groups that have larger home ranges or species that have larger home ranges. Although that is likely to change, you would expect to see intra-specific variation as well that would play into it. But yeah, essentially, a larger home range would mean a greater spatial spread of the group. And more broadly speaking, you can expect to see a more clustered or a substructured social network.
Okay. So the last question is by Aditya, and he wants to know what specific models can be followed to study the primate bird association with seasonal variations.
Primate association with birds, was that the question?
Yeah, with seasonal variations.
But I'm not sure about what that would mean. Would that be related to comparative studies between primates and birds across seasons, or primates associating with birds, or interspecies interactions between primates and birds? So
Okay,
that's
so Aditya, yeah, so Aditya is there. Maybe I can unmute him and he can ask it.
okay. So I
Itself.
see he said it's interspecies.
Okay, yeah.
So in all honesty, I'm not too familiar with interspecies associations between primates and birds. But I do know that there is research that looks at interspecies associations on a more broad level, in the sense that primates coexisting with ungulates like deer, or rather herbivores like deer and so on and so forth, and how they form mutualistic associations. For instance, alarm calls by langurs and bonnet macaques in India have been shown to be used by deer and other ungulates to potentially avoid predators and so on and so forth. So there's research that's again scratching the surface on that, but I'm not aware of in-depth interspecies associations when it comes to empirical research, and particularly when it comes to research that has used social network approaches.
Okay, cool. So I think we are done with all the questions. So there's just one small thing. So Avdut would like to know what is the name of your cat.
Oh, so his name is Vetri. So there's two cats. We have Vetri, and the other guy is Kali. So the guy who made a guest appearance, his name is Vetri.
Vetri. Oh, nice. So thank you, Krishna. Thank you for the wonderful talk.
Yeah, you're welcome.
I'm sure like people enjoyed it. So there are a few. So for the audience, there are a few announcements that we would like to make. So please tune into the AIP channel for future talks. So there is one talk which is coming on the 12th of May. But as I said that there might be change in schedule. So please tune into the AIP channel for future talks. And I would like to thank all the participants for attending this session and for sticking around for so long. Initiate this nice series of talks. Yeah, thank you, Krishna, again.
Yeah, thank you very much. And I just want to restate that it's been a pleasure, and looking forward to a number of more such talks that AIP has taken the initiative to organize. I think you guys are doing a great job, and we
Thanks.
need more people coming and giving talks.
Yeah.
So
And we hope
yeah,
that and we hope that we will invite you again in future for some different talk. Let's see.
yeah,
And yeah, I would also like to thank all the members of AIP who helped in organizing this talk.
yeah. Shout out to everybody. Great.
Yeah. Yeah. Thank you.
All
So
right, guys.